Hello, this is second video of LSCI 5012 for discussing electron tomography of cells. So electron tomography is a 3D electron microscopy techniques to improve resolution of transmission electron microscopy. In the previous lecture, I explained how samples for transmission electron microscopy are generated, and in this video I will talk about a method that increase the resolution of electron microscopy. So there are two parameters that we use to describe performance of microscopy. The first is magnification and the other is resolution. So magnification means how much a microscope can enlarge the images under the lens or on the stage. For example, here in dot in A, it's just a circle, but if we increase the magnification of microscope, let's say it turns out to the dot, let's say the dot consists of 3 H-shaped structure. So if from A to B there is an increase in magnification, and also you get more information by magnification. The reason why you get more information is that the dots inside of this circle now are recognized as separate points. So resolution, it means the short distance between two points that are recognized as separate entities. So by resolving individual points that used to be in this circle are now shown as individual spots that constitute this 3 H-shaped structure. But magnification and resolution always go hand in hand together. Sometimes you can increase the magnification without improving resolution, as shown in the change from B to C. So from B to C, these 3 H-shaped structure are still shown here, and they are separate from each other more because you magnify the magnify the image, but there is no new real information by doing this. So in this case, this is an empty magnification without getting any new information. So I'll explain this situation in more detail using real electron micrograph. This is a bright field micrograph of a maize endosperm. This epidermal cell layer of endosperm is called like basal endosperm transfer cell layer. So this is maternal pedicel, and this is a magnified view of this area indicated by this light blue square. So from transition to here, there is a 10 times increase in magnification, as you can tell by these changes in scale bar. This is so you can see the corresponding area from this cell and the other cells. So this cell wall looks like a line, but here it seems that the cell wall actually have some ingrowth that grows into the cytoplasm. There are a lot of round spots in the cytosol, and then this is a nucleus of the cell. So you can get more information by magnifying this micrograph from this low magnification to higher magnification. But if I increase the magnification 10 times more to focus on this area marked by light blue square in the middle panel, I get this. But I can see kind of matching structure, but I don't get any new information. That I see these dots enlarged view, but they just look the fuzzy, and I don't get any new details around in the cytosol as well as the cell wall. So this is the empty magnification because there is no improvement in resolution. But in fact, I lost a lot of information that surrounding this light blue rectangle. In fact, resolution is limited by the probe that is used to generate image, the wavelength of the probe that is used to generate image. So this is an equation to calculate out the resolution of a microscopy. So this is the d equals the distance between the two dots that are imaged as two separate entities, is calculated by 0.61 lambda, the wavelength of the radiation, divided by n, means the refractive index of the media between the sample and objective lens, and sine alpha, where the alpha is a cone of the angle within which light from the sample is collected. So this n sine alpha is determined by the objective lens that you use, and is given as one number called the numerical aperture. So if I calculate the resolution limit of light visible light microscopy, I should put the wavelength of visible light. The shortest visible light is 400 nanometer, and one of the largest numerical aperture currently available objective lens is 1.2. So this is about 200 nanometer. So there is a theoretical resolution limit of light microscopy which is around 200 nanometer. So to improve the resolution of microscopy, we have to use either increase the denominator or make this lambda smaller. So because there is a limitation in increasing the numerical aperture, the people start looking for probe radiation that has shorter wavelengths. So these are some of the radiations that we use to generate images of cell. First is light. We have I just have showed in previous slide. So light is easy to focus using curved glass, and it's easy to observe because we can just see them with our eye. But the problem is, as I said, it's a longer wavelength. As the X-ray is another radiation. It has a shorter wavelength, and it has a very good penetration power, so you can see the inner structure better. But the problem is X-ray cannot be focused by curved lens, and second, X-ray energy level is about the can break the covalent bond that constitutes the cell, so it could damage the cell. So these are the cons. And third is electron. So electron is a particle, but when small particle exhibit wave-like property. So by increasing the energy of electron, we can make a wavelength shorter. And second, the electron has a charge, and using electromagnet, we can bend the flying path of electron beam, so we can focus to generate image. But the disadvantage is electron cannot fly long distance in ambient air pressure, so we have to image under vacuum. And then electrons also cannot penetrate deep into the tissue, so sample has to be cut into thin slices. And also electron can be damaging too by adding charge and ionization. So, but the people managed to make electron microscope, and we currently use light microscopy and electron microscopy to image cells and the component of the cells. So this is an example of cellular structure or cell that we can see with the naked eye with light microscopy and by electron microscopy. Eukaryotic cells are usually around the size from 10 to 100 micron. So we can see the cells, eukaryotic cells, with light microscopy, but cannot see most of the eukaryotic cell with the human eye, which has a resolution limit around 0.1 millimeter, which is the thickness of human hair. By using a light microscope, we can see individual cells and large structures of cells such as nuclei, chloroplast, and mitochondria. And bacterial cells you can see as kind of like a small dot. But as you have seen, light microscopy has a resolution limit which is around 200 nanometer. So with the light microscopy, you cannot see viral particles or constituent of cells such as ribosomes and lipid bilayer, actin filament, or other proteins. So using electron microscopy, we can see smaller the structures, and we can see many subcellular structures and constituent of even cells and organelles. And electron microscope, biological electron microscopy, we usually have resolution around the 1 to 2 nanometer. So these are millimeter and micrometer and nanometers are units that you have to remember when we talk about cells and tissues, because millimeter we use this to describe size of tissues, and micrometer we use this for cell sizes, and nanometer for this describe organelles and membrane and cytoskeleton. So angstrom is 1/10 the size of nanometer, and this is about size of a hydrogen, and this is used by biochemists. So this is comparison of light microscopy and electron microscopy image of the yeast cell. So this is a phase DIC, normal scale optics of a yeast cell, where you can see the nucleus and you can see the cell and surrounding media. And this is an image of a cell after staining the DNA, showing the nucleus and mitochondria around them. And this is electron micrograph of a similar cell, showing plasma membrane and nucleus, Golgi and mitochondria and cell wall. But we can see more details about the structures of individual organelles. Like nucleus, you can see nuclear membrane and nucleolus, where the ribosomes are made, and then chromatin area. In mitochondria, you can see cristae, and in ribosomes inside of mitochondria. And this is Golgi stack, and you can see stacked Golgi, and one side is lightly stained and the other side is darkly stained. So these things you cannot see in light microscopy because of resolution limit. So we have improved, we can improve resolution by using electron microscopy, although there is electron microscopy has its own problems. So although we have increased the resolution, electron microscope images are two-dimensional projections of 3D structures. So depending on how the object is oriented under electron beam, the structure could be seen in different shape. For example, this column is oriented in parallel with the electron beam. It shows the face top-down view, which is a circle. But if this column is located sideways, and the projected view of this will look like a square. So from the one projected image, we cannot have a full grasp of the structure in 3D. And this is a cartoon that I stole from book illustrating the problem of two-dimensional projection of a 3D structure. This dancing rabbit in front of the beam projector could look like the hand that and the hair, the ear, and the legs of the rabbit could look like fingers of a hand. So if you are interested in 3D structure of a large structure that you can make many slices out of them, like about using ultramicrotomy, you can collect the serial sections and then stack the images in computer or some other way, and you can have 3D information. For example, this circle, if you get this type of image from one end with a tangential section of a circle and go into the center where it's larger and get this thinner, so since you know the thickness of each section, you can have an estimate of how tall this is, and by assembling this, you can have an idea that whether this is like a circle or some other shape. And in B, so this is a bit more complicated. So let's say this is egg. If you get a center section, you will look like this. But by having section from many angles, and then by integrating this information, you will have an idea what this the shape which has the darker nucleus, a core inside. And this is a bit more complicated. So to have a information about this, you need quite many sections in many directions, but in the end you will have an idea what the 3D structure look like. So to do that, you have to do serial sectioning, as I explained on the previous video, where you get a ribbon of a section and then examine the same cell or same organelle in the serial section in the consecutive section in the ribbon, and you assemble this type of image to get structural information in 3D. And this is an example of such analysis. This is a developing C. elegans embryo cell, and these are mitochondria. And so in one image, we can recognize which are mitochondria, but we don't have idea what this mitochondrial organelle. Mitochondria is organized in 3D. So in the movie, about like 20 serial sections were collected and imaged and then made into movie, and each slice of section mitochondria were outlined and to generate 3D object, and by changing orientation of this 3D object, we can have this image showing the 3D organization of mitochondria in this developing C. elegans cell. And this is a mutant where mitochondrial fusion is inhibited to this cell. The mitochondria look like more round, although there is some sausage-shaped one. When compared with this cell, the mitochondrial organization is very different. So mitochondria were outlined in red color and shown here as an individual like bubble. And then by showing this in multiple orientation, we can easily compare mitochondrial organization in this cell. So we can address the problem of 3D problem of two-dimensional projection problem 2-dimensional projection of a 3D structure by serial sectioning if the section the object we are interested in is pretty large. But what if our structure is small and the one section contained in one section we cannot cut even we cannot cut thinner? For example, let's say the previous the column is contained in one section which is about like 80 to 150 nanometer. So there is no way we can do serial sectioning of this. This is a electron micrograph of about 60 and 80 nanometer thin section of a plant cell showing the Golgi and trans-Golgi network and cytoplasmic ribosomes around them. So I will use some of these ribosome clusters. So you can see this series of black dots almost touching each other, but in fact they probably not in contact with each other because these ribosomes are only 15 nanometer. There could be many ribosomes in one point in 2D, but they are actually aligned in different height in 3D. So like here, so this is like 80 nanometer section, and these are 15 nanometer ribosomes oriented in this, located in the section. But from the G, when electron is penetrating the section and made the image, it seems that as if they look clustered and they are touching each other. So this is a problem in electron microscopy. So you may have heard of confocal microscopy, the G-stacking in confocal microscopy. In case of confocal microscopy, you can have a small point in front of light source and in front of the detector. So you can selectively excite fluorophore in certain plane in focus and collect selectively fluorescence coming from the focus plane to enhance signal from the plane in focus, and you can change the height, the position of the focus plane along in the G, and then you can actually get 3D information by changing the position of the focus plane. That is called the G stacking. So basically, you can selectively focus image in a thicker sample and get a better view of it. For example, this is a developing Drosophila embryo epidermis, and then this look like this like cloud in just in regular AP fluorescence. But if you get a focus from about only 1 micron thickness, which is in focus with the light excitation and the detector probe, you get better resolved image like this, and you can collect maybe 20 of this. As you change the height of focus plane, you can get 3-dimensional information of this. But in fact, you cannot selectively focus certain height in TEM sections because everything in the TEM image is in focus, because in TEM we have an aperture in front in the optical path, and the smaller the aperture, the thicker depth of field. So everything in the TEM section is in focus, so you cannot selectively focus out a certain plane in the imaging field. So if you have used a camera with an aperture, you may have seen this phenomenon. I can explain that with this image. So this is a picture of mug cups on the table. So if you have a large aperture, you get light mostly focused on the somewhere in the space. But if you use a smaller aperture like this one, so everything in the field is in focus, although you have to expose a bit longer time. So because everything is in focus, you cannot reject out the light coming from unfocused plane in TEM. So researchers have devised two ways to recovering the information that was lost by collapsing 3D images in 2D. The first is electron tomography, and the other is single particle method. So since single particle method requires multiple copies of same object, so we cannot use this method for 3D analysis of cell, because no cells are same. So we use electron tomography. So electron tomography is basically same as X-ray CTs doctors do in the hospital. So you get an image of an object in multiple angle, and these images from multiple angles are collected, and they are calculated in computer to make a virtual 3D file from which you can extract out images that represent a 3D, the 2D section of the 3D object. So this is the same, almost same slide as previous one. So let's say you have a circle, spheres, and then columns, and you collect these images, and these images are projected back into 3D object, and from which you can prepare images that represent the cross section. So this is a virtual 3D object, and I briefly explained how this could be done. So this could electron tomography is possible because of projection and slice theorem. So this utilizes the Fourier transform. So let's say you have suppose you have this three-dimensional object which is a column, and using TEM you can collect the image of this in multiple angle by turning this, and this individual images of projected view can be made into calculating the Fourier transform, and they are sent to the reverse space, and then you can merge all this in the computer into a kind of 3D object in the reverse space, and then you can reverse Fourier transform back into real space to reconstruct this. So you cannot get information in the real space, but you can combine the 2D projections of you can combine Fourier transform of 2D images generated from projected view of this real structure, and to make a 3D object in the reverse space, and then you can send back to real space to get a virtual 3D object from which you can get a section view of it. So how this could this be done? So this is a real data of electron tomography. So this is an image of a plant cell, where you can see cell wall, endoplasmic reticulum, vacuole, mitochondria, Golgi, and multivesicular bodies. And this is an image from zero angle, and I collected 81 images from plus minus 60 degree with 1.5 degree interval between them. So you can see these images that shows the image of the section at multiple angles, and Fourier transform of these individual images are made in computer, and virtual, and then those 2D images of the Fourier transform of 2D images are combined in the reverse space and projected back into the real space. So how do we collect data? So this is in regular TEM we just put EM grid in the tip of the grid holder and put it in the EM. We collect images, and basically it's same. But the difference in tomography, electron microscopy, is the fact that there is a very sophisticated motorized stage that it can turn the EM grid very accurately multiple angle, and they should have a bit stronger electron beam because the higher the angle, the thickness of section increases, and then you need a stronger electron beam to generate image of a section when it is tilted into higher angle. And also it should have also very accurate XY position controller. The reason is because the EM section is not always located at the center of the rotating axis. So in this image is shown as if the center of rotation is located at the mid position of the images, but in fact this is not true in almost most cases, because section is located off from the rotation angle, and as it moves, its height changes and also XY position changes. So when its position changes, the XY stage has to move so that this go into the same position in the electron beam. Also, it has to be able to change it. Should be able to change the G position to make it in focus, because as the G position changes, the focus changes too. So this is called non-eccentricity, and the theta axis offset. So to solve this the problem, automatic tracking and focusing image should be done, and you might wonder what these small particles in here. So these are gold particles that is used to align the images afterward to get a very well positioned tilt series. After you do all this, and with a computer program that I'll explain later, you can calculate tomogram, and then you will get this series of images that represent that shows the section that are sliced into far thinner by electron tomography. When you compare this image with these individual slices of electron tomogram with this, you can see the increase in resolution by doing electron tomography. And good thing about this tomography is not just increase in resolution. These images are already in like ordered and aligned already. So by scanning through these images, you can get 3D information. So as I said, tomography technique can optically, mathematically slice the volume, so it increases the resolution, because the thinner the slice, the better the resolution. Suppose you have a smiling face in like 100 nanometer thin section, and then you get a very good diamond knife and then cut them into 60 nanometer physically into section. Physically, you get 3 slices. But if you put this 180 nanometer smiling face into tomography microscope and then cut it into 2 to 4 nanometer, you get far more slices. And anything in this thickness is projected into 2D volume. The thinner, the better the resolution. This is what you see in this individual tomographic slice, where the ribosomes, individual ribosomes, these black dots, individual ribosomes, and they you don't see these clustering of this anymore, as in this the regular view of that thick section. So I mean, if I showed this, I mean, you can tell that the improvement better. So this is the same image in previous slide where I showed Golgi stack and trans-Golgi network in the plant cell cytosol, and the ribosomes are all surrounding, all these ribosomes are surrounding the Golgi stack in the TGN. So in the 2 nanometer tomography slice generated from this section, you can see individual ribosomes a lot better. And unlike this image where the ribosomes are closer than touching each other, we don't see the ribosomes clustered that close to each other. And another improvement in membrane outlines. So this TGN looks like a cloud in this image, but you can see outlines of individual vesicle and budding profiles are clearly resolved. This improvement came from the fact that this slice is far thinner than actual physical TEM sections. So, and because the tomography consists of better resolution images that are already aligned and ordered, by scanning through the individual images, we can get 3D information. Suppose you have the smiling face and frowning face in the tomography EM section. They will look same if you just get a image with a regular transmission length microscope, because you cannot tell whether this mouth is curved up or down. But you break this section into 2 nanometer thin sections by tomography, you can collect images from the spanning the mouth 1, 2, 3, 4, 5, 6 images, and then carefully examine whether this the mouth area to tell whether this mouth is curving up and curved down to tell whether this face is smiling and frowning. So usually we do not have a smiling face in the cell, so this is what we usually get. So these are 3 consecutive images from the serial the tomogram of the Golgi stack. So when you carefully examine these outlines of, for example, this vesicle, this outline of this vesicle changes as we move from A to B and B to C. So we can see there is a change in 3D-dimensional organization, but it is a bit difficult to tell how that changes in 3D. So to better visualize this 3D organization of membrane and the ribosome excluding zone surrounding the Golgi, we generate outlines in 2D, and these 2D outlines are combined in computer to generate 3D object. So this outlining used to be done by hand, but nowadays image recognition and pattern recognition algorithm gets better in computer, and this could be like automatically or semi-automatically. So this is a 3D model. It's the Golgi, and we can turn this Golgi model around to visualize this 3D structure in multiple angle. But the merit of generating 3D model is not just the visualize purpose. We can actually calculate out morphometric parameters from the 3D object. For example, in this trans-Golgi network, some area of the membrane is colored in green. So this area in the TGN corresponds to the segment of the membrane of which curvature matches radii of secretory vesicle, suggesting that these are the area in the TGN where secretory vesicles are budding out. Since the secretory vesicle does not have very obvious coat, such as coat as like clustering coated vesicle, it is difficult to where the vesicles are budding out. So this is one way to tell the candidate area where the secretory vesicles are budding from the trans-Golgi network. So we can calculate curvature of the membrane. Also, we can calculate volume or surface area of the membrane structure. So this is another example that morphometric, the quantitative information was extracted from 3D model. So in this blue cisternae are trans-Golgi network identified by association of certain molecular marker, and this magenta colored cisternae are the transverse Golgi cisternae, and there is about like 30 to 40% surface area reduction when we compare surface area of the transverse Golgi cisternae and trans-Golgi network cisternae trans-Golgi network compartment. So we can calculate surface area very accurately from this 3D model. So by adopting electron tomography, we can make quantitative prediction or comparison of high resolution structure in the cell. And this electron tomography technique could be combined with electron immunogold labeling. So you may remember that immunogold labeling is a technique that is used to localize macromolecules using antibody in electron microscopy. So we can localize particular epitopes in the on the surface of EM section very accurately by combining the location of the immunogold particle with a 3D model of the generated from electron tomography technique. So for example, this is localization of monocytase 1 in Arabidopsis, and gold particles are shown here as yellow spheres. And then this is overlaid with the Golgi model generated from the tomogram of this Golgi. And when we count the number of gold particles associated with each cistana, we found out that most of the gold particles are associated with C3 and C4. So indicating that this antibody, the epitope of the monocytase 1 is mostly located to C3 and C4, which is medial Golgi. And this is 90-degree rotation of the same model, and the gold particles are aligned in certain location. The reason is because gold particle cannot go into the section, so these are the surface of section where the gold particles are located. So this suggests that monocytase 1 is the first enzyme involved in N-glycan modification in the Golgi, and then N-glycan modification begins from the medial Golgi, not from the cis Golgi. So so far I've explained the basics about electron tomography. So from this I will talk about more of a practical aspect of electron tomography. So to do electron tomography, we use a software package called iMod. So iMod is a set of command that you can use in the Unix terminal window and to calculate tomogram and for processing and the modeling of the organelles in tomogram. And this was developed by 3D electron microscopy laboratory of the University of Colorado at Boulder. And this is a free program, so you can go to their website and download them. So they make three software for the three different platforms. So you can run this in the Windows or Macintosh computers, Apple computers, or Unix computers. So for the Macintosh computers, Apple computers, and Unix, you do not need any extra the installation because they are running on Unix background. So you can just install the program. But in Macintosh computer, you should use terminal window shown here, where you can type the command line command. So and for Windows computer, you have to install the Unix emulator such as Cygwin, and then once you install Cygwin, you can open terminal window, and then you can use the iMod software package. So in iMod software package, there are two big programs compared with other commands, which are eTomo and 3D-Mod. So eTomo is a graphic user interface for the common processes like tomogram calculation. In earlier stages of iMod development, so you had to type individual command with their parameters one by one in the terminal window, but sometimes you do the same thing over and over when you calculate tomograms for like serial sections. So it's a bit cumbersome to type all these and repeat all the time. So by having eTomo graphic user interface, you can just press a button with your mouse instead of typing, so which make things a lot easier, and also you can kind of semi-automatize tomogram calculation if you have a lot of data set. 3D-Mod is also graphic user interface for generating 3D model from tomographic slice images, and also this is a software you can make a movie out of tomograms, or you can combine model views and tomography slice views, and also get morphometric parameters and such as like densities or the surface area of the structure. And this is a start window of eTomo, where you can use to build tomograms, or you can, sometimes you have to join tomograms from serial section and then stack them into Z direction, so you can join them together, and also you can do other things with the eTomo graphic user interface. So if you go to the website bio3d.colorado.edu, so you can see what it looked like. So this slide was generated some time ago. Now the version is like 4.15 or something. But this particular the screenshot was taken when the most current software version was 4.7. So this is a window where it shows a line command of that runs on Unix platform in iMode. For example, like iMode mesh. This is a command that you use to generate a 3D surface out of two-dimensional contour. So you can use this command. And if you each title has a hyperlink. So if you press this command, you can go to the web page that explains how to use it, and the history of the command and how to use this command. And they grouped into model utility, image processing, and other things too. So there are quite many of these, and if you are interested, you can study this individual command. But as I said, typing individual command in Unix terminal window is not so convenient. They developed eTomo graphic user interface for common procedure. So this is a window showing the building tomogram. So if you open eTomo, then press build tomogram, it prompts you with a setup window where you type in basic information about the tilt series. Then if you finish that, you go to this window where you can do like a tomogram calculation of A-axis and calculate tomogram in B-axis, and then you can combine them. So if you press this first step where it's called pre-processing, you came up with this type of window where you can put some parameters and press like this button to find out X-ray, like the spots is oddly like a bright that may correspond to the X-ray, the spot generated not by electron but by X-ray. So you can, those things could be like fixed by this pre-processing, and you can do this and go to like coarse alignment and then fine alignment and tomogram calculation. Once this could be done, this was done in both A and B axis. A and B axis can be combined to generate dual axis tomogram. So this is eTomo window. So this will be explained in another separate video with the example data set in the course. So once you finish the tomogram calculation, you can generate 3D object. So this is a window you get. I mean, I opened all the window, but if you open 3D mod, another big program in the iMode to generate 3D model from tomographic slice, and then get a specialized view like tumbler view or isosurface view, and you can control individual objects such as color and like a shape, or they're like a texture, or those things could be controlled in this. So this is a 3D mod window to generate 3D object and also movies of the 3D object and tomographic slice. So if I explain 3D model generation in a nutshell, so the most important concept in 3D model generation is hierarchical structure. So 3D model is in 3D, and there could be one model can have many 3D objects. So object is one closed surface, like a membrane compartment in 3D. And this three-dimensional object consists of two-dimensional closed that polygon. So this contour is outline of a membrane compartment or like cytoskeleton in 2D. So this is made of line, and then the point is like a point. So the two-dimensional contour is made of points. So when you draw outline of a membrane compartment, you put point. Instead of a point, constitute contour. And this contour should be in one tomographic slice in case of membrane compartment. And these contours in each slice constitute the 3D object. And then 3D object in a one group makes a model. So this is how you generate model, and this is show the model layout. For example, objective 1, as I said, made of many contours in 2D. So you can see like 3 contours, and each contour there are substructure that which corresponds to points. Contour 1 has 2 points, and then object 1 has 3 contours, and contour 1 has 2 points, contour 2 has 3 points, and contour 3 has 2 points. So the same thing like object 2, object 3. So this is how you generate a 3D object. So this is a tomographic slice of a Golgi where I outlined one transverse Golgi cisterna with dark blue line. But this dark blue line in 2D image has many points here, and in here you can see like red circle and green circle and yellow circle. So green circle means the start point of a contour, and red circle indicate last point of a contour, and yellow circle marks the point that I selected. So in each slice I make this 2D contours, and then I repeat that in many slices as shown here. Now I change the tomographic slice and draw outline one by one. And this two-dimensional contours shown here is meshed into 3D surface, and this is one object. So this iMod mesh is a command that changes contour data into 3D surface by meshing. So, and then I can do this for many cisternae, like not just transmos, cismos, then C2, C3, C4, C5, and C6, and then trans-Golgi network. And I usually do it in different color. And then I repeat all those, and then they can made it into this 3D structure, and then they are grouped into one Golgi model where I can see cismos and trans-Golgi network, and transmos cisterna, and C2, C3, C4, C5. So this is how you generate model, and this model generation part will be demonstrated in the separate class, and then you will have a chance to draw outlines and make mesh to generate 3D model in the practical sessions during the course. So this concludes the lecture about basics on electron tomography and the software that we use to generate 3D model and make tomograms. Thank you.